So now we need to talk about CRISPR-Cas9. CRISPR-Cas9 is a new technique that researchers are using to splice in vivo in live cells. Actually in an organism, they're splicing new DNA into chromosomes or other DNA in the genome. The CRISPR system is capable of actually editing a chromosome. It's capable of cutting out a bad section of DNA and replacing it with a good section of DNA. CRISPR actually stands for clustered, regularly interspaced, short palindromic repeats. Basically, CRISPR was discovered in bacteria. Oh, by the way, Cas9 stands for CRISPR-associated protein number 9. I'll get to that in a little while. CRISPR is basically part of a normal bacteria or archaean immune phenomenon. It turns out that bacteria are constantly being attacked by viruses, and bacteria need to protect themselves against the viruses. So the way bacteria do that is they use CRISPR. A CRISPR DNA section actually consists of repeating DNA sequences of genetic code interrupted by little spacers. When researchers discovered these in the 80s, they didn't know what to make of them, and they didn't know what they were exactly for. In the early 2000s, it was proposed and initially rejected. Many times when new theories are proposed in biology, people blow that off. And then now the people that did that with CRISPR are being silent because they realized they were galactically unintelligent. CRISPR is a system that bacteria use to protect themselves to virus. So here's how it works. A bacteria is attacked by a virus. What happens is the virus injects its DNA into the bacteria. What the bacteria does is, with its restriction enzymes, it cuts up part of the virus and it actually stores the virus inside of its genome in the CRISPR region, the CRISPR gene region. The reason for that is if the virus attacks the bacteria again, guide RNA is manufactured. This is weird. It's called guide RNA. It's not messenger RNA or transfer RNA or ribosomal RNA. It's guide RNA, which is made from the CRISPR regions. The guide RNA will then attach to the viral DNA and mark it, so then a special Cas9 enzyme will then cut the viral DNA up and destroy it. So basically what bacteria are doing with this CRISPR system is they can remember the infection of a virus by storing part of its genome to engage further attacks to that same kind of virus when they get attacked in the future. It's kind of an ingenious system. Researchers realize that we can actually modify this system to do our own DNA cutting. By modifying the system, you can use the CRISPR system to go into a human cell, go into the chromosomes, bind to a specific region of DNA, cut out a bad region of DNA that you want to remove, splice in a good region of DNA, and then seal it in like with a ligase. This has never been possible before. And we are on the verge of curing genetic diseases like muscular dystrophy and cystic fibrosis. It appears that the muscular dystrophy research team is in the lead, and they'll probably be working on the first human trials within the next 1.5 to 2 years.